side. I'm going to be pointing at stuff like this. If you're over there, you can't see what I'm pointing to. All right, so what do we have forming the surface of the skeletal muscle cell? What do we have forming the surface of the skeletal muscle cell? What do we call the cell membrane of a muscle cell? The sarcolemma, exactly. So this is the sarcolemma, the skeletal muscle cell. This is the sarcolemma of the skeletal muscle cell right here. So you'll notice that there are little holes. There's a hole in the sarcolemma. There's another right there. Same thing here. There's a hole. There's a hole. What are the holes lined up with? Follow the hole inward to this gray tube going in. Follow that hole inward to this gray tube. In this case, it's a yellow tube. So there's the hole, the yellow tube going in, the yellow tube going in, the yellow tube going in. These yellow tubes are called transverse tubules. It's gray transverse tubule, gray transverse tubule, yellow, yellow, yellow transverse tubule. Each transverse tubule is an invagination of the sarcolemma. So the sarcolemma is actually extending into the interior of the muscle cell. So it's actually a tube of sarcolemma extending all the way in to the interior of the muscle cell. Let's see. The rest of the yellow, I should have used two colors the way this model did, gray and blue, because the rest of the yellow is a completely separate object called the sarcoplasmic reticulum. So there's a specialized, smooth endoplasmic reticulum inside muscle cells called the sarcoplasmic reticulum. Remember, the cytoplasm of muscle cell is called sarcoplasm. So the sarcoplasmic reticulum, not the tube, but this bulge here, this bulge, this network, bulge, network, bulge, network. The entire sarcoplasmic reticulum permeates the interior of the muscle cell, and it stores calcium ions. The key function of the sarcoplasmic reticulum is to be a calcium ion repository. So, what happens is, let's say we start an accidental right here in the sarcolemma. We'll deal with how that happens later. So just work with me. We start an accidental here in the center of the sarcolemma. In the case of a muscle cell, that's where it starts. Not at the end. With a neuron, we start at the initial segment of the axon. With a muscle cell, the accidental begins in the center of the sarcolemma. So instead of going one way, it actually goes both ways. So start, whoops, I took off the whole peg, start an accidental, it spreads simultaneously away from that point in both directions. So, let's see, what's that? Oh, that was glued on, is no longer glued on. Okay, so much for that. So the accidental spreads, gets to a transverse tubule, and the accidental continues internally along the transverse tubules. Why? Because there are voltage-gated sodium channels in the sarcolemma surface, and the transverse tubule membrane continues the voltage-gated sodium channels. So the accidental goes all the way in, all the way in. Look at these bulges of the sarcoplasmic reticulum wedged against the transverse tubule. They're called terminal cisternae. So terminal cisterna, transverse tubule, terminal cisterna. Same again here, same again here. That triple structure, cisterna, tubule, cisterna, is called a triad, a three-part structure. Cisterna, tubule, and cisterna. Okay, what's significant about the cisternae is that they are wedged against the transverse tubule, they're here in blue here, terminal cisterna, transverse tubule, terminal cisterna. They're wedged against the transverse tubule, and the cisternae contain voltage-gated calcium channels. The accidental starts here, spreads simultaneously in two directions away from here, continues down the transverse tubule, and continues out over the terminal cisternae, opening up voltage-gated calcium channels. So calcium ions come diffusing out to beat the band. There's packed in there in such high concentration As soon as you start opening up calcium channels, calcium comes diffusing out at rapid pace. So, we'll talk about the role of the calcium shortly. Other goodies to point to here. What's this thing? What's that thing? Same thing. This is a mitochondrion. Mitochondrion for aerobic respiration. What are these little dots here? These little orange dots. These little red dots, again, same thing. Glycogen granules. So skeletal muscle stores granules of glycogen. Glycogen is sometimes called animal starch. It's the characteristic energy storage form of polysaccharide in animal tissue. Glycogen is. It's one glycogen molecule is a long, convoluted chain of glucose molecules. So it's the energy source for aerobic respiration and the mitochondria. So a mitochondrion and a couple of glucose granules. Mitochondrion and some glycogen granules, beg your pardon. Let's see, this log-like object here is one myofibril. Here's another myofibril. Notice that they are side by side, so cross-sectional view, there's one myofibril, there's another myofibril. And this cross-sectional view reveals little dots on here. Unfortunately, the dots are all the same damn size. So that must mean we are going through, that's why they're all the same size. We're going through just the thin filaments. That explains it. So, we're just going through. Look here. These are thin filaments. There's a Z disc. See that zigzag pattern? That's a Z disc and thin filaments. So thin filaments extend from both sides of a Z disc. So this cut is just through thin filaments. That's why all those dots are the same size. This cut is through thin filaments and thick filaments. See the difference in diameter here, partially overlapping, thin and thick, right there. So this cross-sectional view, I'll have dots of two different sizes, and voila, we do. The bigger dots, cross-sections of thick filaments, the smaller dots, cross-sections of thin filaments. So that would indicate an area where the two are overlapping one another. And again, myofibril. So what do we have here? What is this section from there to there called? That's an I-band right there. A Z disc runs down the center of an I-band. So there's one complete I-band. There's one complete A-band right here. And we start another I-band, etc. So I-band, A-band, I-band, A-band, all the way down the length of the myofibril. Let's see, what else we have? Oh, this one properly shows that a transverse tubule comes in at the ends of an I-band. So this transverse tubule is coming in at this end of the I-band. This transverse tubule is coming in at this end of the I-band, right there. So that's correct placement. Why am I missing that? Because we get it wrong on here. Because here we've got 1, 2, 3 myofibrils within one skeletal muscle cell or myofiber here. Transverse tubule, terminal cisternae, all of the blue represents the organelle called the sarcoplasmic reticulum. But they're showing the transverse tubule coming in along the Z disc. So there's an I-band, that's supposed to be a Z disc. There's another I-band, that's supposed to be a Z disc. There's an A-band right there, a bundle of thick myofilaments. There's the M line, and that gray portion in the center, that's our H zone. So A-band, H zone, M line. I-band from there to there with a Z disc running down its center. So what this model gets wrong is it has the transverse tubule coming in lined up with the Z disc. The reality is the transverse tubules come in lined up at each end of an I-band. So not here. This model has the transverse tubule coming in along the Z disc. Instead, they come in at the edges of the I-bands. Okay? Anything else? Any